This is Professor Salman. We're really delighted to have him with us for our second journal club. So he's going to give a short presentation, and then we'll have a Q&A after that. And some people have submitted questions, and I'll ask those on people's behalf. But if you have any other questions, you can message me directly, and I'll try to fit those in as well. And Professor Salman is the Professor of Clinical Neurology at the University of Edinburgh. He studied at Cambridge, and then he trained in Edinburgh as a clinician scientist working in the stroke research group. He's also recently become the lead of this group, and it's been renamed to the Cerebrovascular Research Group, and it consists of a number of ongoing trials and I believe around 10 PIs. And his research are mainly in frequency, pathophysiology, treatment, and prognosis of intracerebral hemorrhage. So I'll hand over to you.
Well, thanks very much indeed, Robin. It's really kind of you to invite me, and you don't need to tell me about NANSIG because I know about all of you, and I think it's a great initiative, and well done for taking advantage of people's availability nowadays for short Zoom meetings. I think this is going to be the new future of the world. But I'm not thanking you for asking me to follow Ian Roberts' act, because he's surely a very hard act to follow. Done some fantastic trials, many orders of magnitude larger than the ones I've been involved in, with practice-changing consequences. But I think this will be a nice contrast for those of you who've already heard from Ian Roberts about the CRASH trials. So I'm going to tell you today just a little bit about the Restart or Stop Antithrombotics randomized trial, or RESTART for short. And before I kind of go into the details of the trial, I think it's really important to say that a lot of the fundamental building blocks of this trial were put in place by medical students. And credit to all of you for being interested in academia and pushing, back the frontiers of knowledge whilst you're students, because many helped me with this. So from medical students who did systematic reviews of the risks of vascular events after stroke due to intracerebral hemorrhage, or ICH for short, as it's put on this slide, to medical students who did systematic reviews of the randomized control trials that have ever been done into intracerebral hemorrhage to try and learn from the methods and the findings of those trials. So kind of meta research, really meta epidemiological research. To medical students who've done the Cochrane review of this randomized control trial and others that need to be added to it to really generate definitive knowledge. So throughout this project, medical students have been involved. So many thanks to all of them. So I started trying to answer this question, I think about 10 years ago. The idea has been around for a very long time, and this is a common dilemma in stroke medicine. We see a patient who comes in with a stroke, a hemiparesis, for example. We scan them, and there's an intracerebral hemorrhage. And this person had already been on antiplatelet therapy, let's say, because they'd had a myocardial infarction a few years before, only now to suffer an intracerebral hemorrhage. So of course we stop the antiplatelet therapy, but as the patient recovers, we know that these patients have a decent, sizable risk of ischemic vascular events as well as recurrent intracerebral hemorrhage in the future. So the question is, should we start or avoid a drug like aspirin or clopidogrel for these patients who've got a good reason to be taking it, like a myocardial infarction or a previous ischemic stroke? Clinicians have struggled with this for years. You'll find review articles decades ago asking this question, and only I was foolish enough to try to do a randomized control trial to answer it. And this randomized control trials built on the systematic reviews I told you about the risks of ischemic events and hemorrhagic events, showing that they are more or less equal, and probably there's an excess of ischemic events after intracerebral hemorrhage in comparison to recurrent bleeding. So that's why we should be concerned about lowering those risks. And there have been observational studies of patients like this, simply observing what happens when antiplatelet therapy happens to be started in clinical practice or avoided. And of course, we as clinicians back winners, so you can't rely on those studies to give you information about treatment effects. You have to do randomized control trials, with all but a very few exceptions, to learn about whether treatments work or not. So all we can say from observational studies comparing apples with oranges is that starting antiplatelet therapy in those studies seems to have been associated with a lower risk of occlusive vascular events, as you might expect, and no difference in hemorrhagic events, which is reassuring. So we did a systematic review before trying to seek funding for this trial, and there were no such randomized control trials, so we got on and sought funding for one. So the design of ReSTART was really aiming to estimate the effects of starting or avoiding antiplatelet therapy with drugs that were used in everyday practice, so aspirin or clopidogrel. It was a very pragmatic trial, so it wasn't blinded. It was just of these drugs in everyday that were available in everyday practice, so there wasn't one agent. You could use aspirin or clopidogrel or dipyridamole, or combinations of them according to the patient's need. We were firstly aiming to look at the safety of these drugs. It would have been nice to have gone for thousands and thousands of patients in a trial to establish the overall effects. But what if we had shown with 500 or 1,000 patients that actually these drugs were harmful? You wouldn't want to carry on looking for overall benefit if you first found that they were harmful. So this was really a safety phase, or a phase 2 type trial, to look at whether antiplatelet therapy might increase the risk of recurrent hemorrhage, and whether that increase in risk might outweigh the beneficial reduction of ischemic events that we know antiplatelet therapy confers. So we aimed to recruit 720 patients in the UK at more than 100 hospital sites. This was a lot of work getting this trial set up, follow them up for 2 years. And we thought, with estimated event rates of recurrent ICH from systematic reviews of between about 2% and 7% per year in observational studies, and a notional relative risk of recurrent intracerebral hemorrhage with antiplatelet therapy between 1, no difference, up to 4, a 4-fold increase, this trial with this sample size might show something that would be clinically meaningful. So this is what we did. We looked for adults who were already taking an antithrombotic drug, so that might have been an anticoagulant or an antiplatelet drug, and that was for the prevention of occlusive vascular disease. So we're not talking about people who were taking them for primary prevention, an aspirin a day keeps the doctor away type idea, because it's still not clear that that's the right strategy to use. This was people taking these drugs largely for secondary prevention, who discontinued them after an intracerebral hemorrhage, as you would, and who survived more than 24 hours, which a lot of patients unfortunately do not after intracerebral hemorrhage, and who therefore, when they're recovering on a stroke unit or when they're seen in outpatients afterwards, have this dilemma about whether to start or avoid antiplatelet therapy. Of course, we love technology in clinical practice, and we use brain MRI, but often we don't know what the biomarkers of small vessel disease that we see on MRI mean. But we like to guide our treatment decisions on the basis of them, but without having ever researched the effects of treatment in randomized trials with those new biomarkers that our technological improvements endlessly demonstrate for us. So we tried to reimburse people with research funding from the British Heart Foundation that funded this trial to do brain MRI in participants who consented before they were randomized, and then randomize them 1-to-1 to start or avoid antiplatelet therapy. We followed them up for at least 2 years, and that was the original intention, but in fact it wound up being at least 6 months and up to about 5 years. And then we stopped the trial. So this is the primary outcome of recurrent intracerebral hemorrhage. And you see 2 groups here. On the x-axis, we've got the follow-up time in the trial, up to 4 years. And on the y-axis, we've got the risk, the cumulative risk of the primary outcome, recurrent symptomatic ICH. And of course, our expectation was that the group of people who started antiplatelet therapy, and there were 268 of them, as you can see below the x-axis, might have a slightly higher risk of recurrent intracerebral hemorrhage, and those who avoided would have a lower risk. And the question was how big this difference was. But to our huge surprise, actually, it was the people who avoided antiplatelet therapy, who had more bleeds, 23 to be precise, than people who started antiplatelet therapy, 12, almost half the number of people who'd had recurrent primary intracerebral hemorrhages in the avoid group. And this was almost a statistically significant reduction of recurrent hemorrhage with antiplatelet therapy, quite the opposite from what we'd expected. So this was very far removed from our prior expectations about the effects of antiplatelet therapy, which generally, in randomized trials that have been done in the past, in people without intracerebral hemorrhage, increases their risk of bleeding again by about 2/3. So in comparison to a hazard ratio of 1.66, what we found was very significantly different from it. So, of course, this result was so different from what we previously expected that we didn't believe it. So we asked the statistician to check and recheck the findings, and of course they were true. You could tell that from the adherence in the groups. This was an open trial, so we could see who was getting antiplatelet therapy and who wasn't. We got the groups the right way around. We looked at differences in blood pressure control between the groups, because we know that lower blood pressure after intracerebral hemorrhage reduces the risk of recurrent stroke, but there was absolutely no difference. The median systolic blood pressure between the two groups was 130 millimeters of mercury. The adherence to the allocated strategy of start or avoid antiplatelet therapy was pretty good, actually, for a group of patients who are on average about 75 years old. And we looked at subgroup analyses, did hemorrhage location make a difference, age, and so on, and there was no difference between any subgroups in this overall finding. It looked more or less similar in all subgroups. So those are the clinical subgroups, and the main thing to say is you can tell this is a trial of 537 patients. This just goes to show how many thousands of patients you need to get really significant tight 95% confidence intervals and be able to show differences between two subgroups. For example, at the top here, by hemorrhage location, a suggestion there might be a difference, but we couldn't show it statistically significantly. But the bottom line here is that you see the overall effect in the diamond, and all of the subgroups, all of the individual subgroups are showing an effect towards reduction of recurrent hemorrhage. So the direction is the same in all the subgroups. You haven't got any subgroups that are going in the opposite direction, favoring avoid antiplatelet therapy. So it's all looking pretty consistent, which is reassuring. We also looked at these biomarkers on brain CT and brain MRI that everyone loves to nuance their treatment decisions by. And the bottom line here, again, without going into detail, is that we didn't find any difference in effects according to subgroups. Again, pretty much all of the treatment effects were to the same side of one, in other words, towards a reduction of recurrent ICH. And in particular, looking at cerebral microbleeds on the right, which was our primary pre-specified imaging subgroup analysis, about halfway down the MRI table on the right, you see that the presence of 2 or more microbleeds was associated with a reduction of about 70% in the risk of recurrent ICH. And this was the group that people would believe might be harmed by antiplatelet therapy. So again, we're showing seemingly quite the opposite. So I'll try and wrap up because we've only got about 9 or 10 minutes left. This was a really hard trial to do. The dotted line is what we said to the funder, the British Heart Foundation, we would do. And the red line is actually what that was our recruitment rate. And that's because people were really reluctant to randomize patients. They often believed they knew what the right thing to do was. And they only randomized 1 in 12 of the eligible patients they came across. So it's very difficult to persuade people without randomized evidence that this trial was a good idea. And I think the reason for that was because we are inherently hemophobic. We're afraid of blood. We don't want to expose people to interventions that have previously been associated with bleeding. But look at how you should question our prior beliefs on the basis of this finding. It's not always the case that antithrombotic drugs may cause bleeding. And in fact, the underlying mechanism under bleeding might be thrombosis or inflammation, which of course antiplatelet drugs affect in the first place. So the mechanisms of recurrent hemorrhage might be quite different from first ever hemorrhage. We tried to use data science to boost recruitment by identifying eligible patients from our collaborators' audit data, but that didn't change the recruitment rate of the trial, as you see in this stepped wedged randomized trial we did as an experiment in trial methodology embedded within Restart. But never mind, we got a really interesting result in the end. You've probably looked at the publications. There's some useful information on our website interpreting it. And we did a survey of what people thought afterwards. And the survey on our website of approximately 114 doctors, 83% said it would change their prescribing patterns, and 90% said they were more likely to prescribe antiplatelet drugs with this result. But would they recruit to a new randomized trial now of antiplatelet drugs for any hemorrhage survivor to test the hypothesis that antiplatelet therapy might just reduce recurrent ICH because of maybe this new insight into the mechanism of that? And more than 2/3 said they would. So that's why we're continuing to follow up the trial cohort and monitoring adherence, seeing if lots of people switch to take antiplatelet therapy. We're looking in more detail at this suggestion of what the mechanism might be from the finding of this randomized trial. And what I'm trying to seek funding for now is a much bigger randomized trial called Aspiring, looking at antiplatelet therapy for prevention of all vascular events, including recurrent hemorrhage, in all comers globally, in Europe and China and other parts of Asia. I won't go into the rationale behind that, but the design is going to look very, very similar, hopefully in about 4,500 patients. So thank you very much indeed for your attention. It's great to celebrate what the UK achieved at all these many sites in Restart. And I can't thank enough all the many people who helped me with the trial. So that's it from me. Back to you, Robin.
Amazing. Thank you. You've actually answered a lot of the questions that came in already, especially about the future and what the underlying mechanism could be that's actually mediating this. I think another recurrent question that came in was about when to actually start antiplatelets, because it seems like it was like 76 days, the average or the median, until people started it from their actual event. And there was quite a big range there. So do you have any idea of when the optimal time is, or do you think that's quite important? Or
So one of those subgroups that was tucked away on that subgroup analysis plot was an evaluation of the effects of antiplatelet therapy in the 50% of patients who were randomized less than the median time to randomization, which was, as you say, a little over a couple of months, versus those people who were randomized more than the median time. And there was absolutely no difference in the effect size between those randomized early versus those randomized late. But as your question correctly implies, it would be nice to have a bigger trial to look in more detail at that, to look at very early restarting versus, a couple of months versus much later. And if Aspiring gets funded, we'll intend to do that.
Okay, amazing. And I guess there's always a question generally from medical students and young doctors interested in pursuing a career in academic medicine. And, the goal, I guess, would be to be able to be in your position to run such a large scale trial over so many years, 122 hospitals. Do you have any advice for young academic clinicians, how they can get involved in this, how they could potentially pursue a future where they could be involved in such trials?
Yeah, really good question. So I was inspired about randomized controlled trials whilst I was a medical student learning epidemiology, which is often quite a dry thing to seem to learn as a medical student. But if you see the questions emerging in your everyday practice as a student, you try and investigate what the evidence is behind them, and you learn the science of randomized trials. It's often quite inspiring, especially with the amount of research that's going on these days, to get involved. So I'm hopeful, and this is something I'm clinical director of the clinical trials unit here. I'd love to be setting up in future apprenticeships for people who are keen to get involved in trials so they can get some experience of how they work at an early age. And that will put many people off because for a seemingly really simple experiment, clinical trials are an extraordinarily hard thing to do. So definitely get involved, learn the first principles, the basic science of randomized trials early on. So doing an MSc in epidemiology via, for example, the London School of Hygiene Tropical Medicine, or the MSc in clinical trials at the University of Edinburgh, or other MScs in epidemiology, is absolutely a fundamental building block for you to learn the science of epidemiology and the design of trials to really equip you well to be eventually running a trial of your own. I started doing clinical research when I was the equivalent of a specialty trainee in 1998. And it took me it's taken me from then until last year to produce my first major randomized trial, which is Restart. There are others that I've contributed to and that are going on. So it takes that long. And I spent a long time doing observational population-based epidemiology, which is a good way of observing uncertainties in practice, collecting data to inform the design of randomized trials, and then doing them. So I think, bottom line, you need to have been around the block quite a few times before actually embarking on a randomized trial of your own. But there's a lot of useful learning and preparation you can do through your career on your way to doing that.
Amazing, thank you. And I think someone's messaged a question, actually. I don't know if you've seen it.
Yep.
Was any neurocognitive data captured as 60% lobar hemorrhages? Does aspirin help prevent this?
Yeah, thanks. That's a good question. So we didn't collect cognitive outcome because, like the recovery trial that's going on with COVID-19 at the moment, this came from the school of simple pragmatic trials. So we really wanted to collect information about these elderly people in a light touch way. And it's pretty difficult for your average 75-year or 80-year-old to complete an annual questionnaire and let you know whether they've had a heart attack or not, let alone testing their cognitive function. There are telephone ways of doing that these days, and that might be an interesting thing to do in a sub-study in a future randomized trial. But we don't have any evidence that there was an effect on that. And do you think that relatively aggressive BP lowering and Restart may have interacted? So I think that what tends to happen in trials is that people are often given excellent care. So patients may get a bit more attention than normal. That's not really the case in Restart because we just left patients in the hands of their GPs and followed them up that way. But there was no evidence that there was a difference in BP lowering between the two arms. And overall, the extent of BP lowering to 130 systolic on average was about what one would expect in everyday practice. So I don't think it interacted.
Perfect. Well, I think we're running out of time a bit, but that was really useful. Thank you so much. And yeah, we're probably going to put this on YouTube again. We're going to alternate between neurosurgery and neurology topics. So the next one should be on neurosurgery. And yeah, thanks again very much for joining us, Professor Salman. And I hope everyone enjoyed this.
Yeah, well, you're very welcome. And sorry that I didn't run quite fast enough to join you earlier and allow more time for Q&A, but be happy to join another one of these in future.
Great. Thanks again. All right. Bye-bye.
Bye.